In the last lecture module, we saw how the combination and interaction of neurotransmitter ligand-gated ion channels and voltage-sensitive sodium ion channels can work to create an ionic cascade that increases both the amount of depolarization and the area of depolarization on a membrane. We saw that the cell body and dendritic branches can serve to sum the activation from all of the synaptic connections for a particular neuron at the axon hillock. Once that axon hillock gets depolarized by about 10 millivolts, that then triggers the action potential which travels down the axon. Now in this diagram we can see how that action potential going from the resting or homeostasis level of negative 65 millivolts up to the full action potential of around plus 40 millivolts. We can also now understand how it is that the action potential propagates itself down the length of the axon by recruiting other sodium channels through voltage-sensitive activation. The action potential, because it is so dramatic, can cause itself to be replicated as it progresses down the length of the axon. But a couple of aspects of the diagram still need to be made clear for us. First of all, why doesn't the action potential propagate in both directions? Why instead do we see trailing shortly behind the action potential and the sodium influx a potassium efflux and a restoration of the membrane potential to the homeostasis resting potential of negative 65 millivolts? What mechanism causes that to happen? Likewise, we'll want to understand why it is that there's this interval of about 6.5 milliseconds between action potentials trailing down the axon here. So let's take each of those things in turn. Why is it that the action potential goes down the axon and not in both directions? What causes that potassium efflux and the restoration of the resting potential? Now, part of the answer here is just timing. When we have that depolarization, the voltage-sensitive sodium channels open up rather quickly in comparison to the voltage-sensitive potassium channels. The same depolarization causes both of the channels to open, sodium first and then potassium. This stutter-stepped or staggered opening of the sodium channel followed by the potassium channel may not seem like a big difference, but in fact it's sufficient to dramatically change the impact that each of these channels has on the membrane potential. When the sodium channel opens, the membrane potential is at negative 65 millivolts, the resting potential, or close to it. Sodium then rides the electrochemical gradient into the neuron quickly. As it does, it decreases the electrochemical gradient, thereby slowing its sodium influx. Once the depolarization becomes sufficient, then the inactivation gate closes on the sodium channel and no more sodium will come in. The slower to open potassium voltage-sensitive channel opens up when the membrane potential has changed from negative 65 millivolts resting potential to positive 40 millivolts or close to it. As a result, the combination of high concentrations of potassium inside the neuron and that positive 40 millivolt membrane potential creates a reversed electrochemical gradient that pulls the potassium out, causing potassium efflux, which will result in repolarization of the membrane at that point. So now we see that membrane depolarization that triggers increased potassium conduction in the form of potassium efflux, which results in membrane repolarization. Now, that potassium channel isn't the only mechanism to achieve repolarization. Remember that we have that sodium-potassium pump, and it too kicks in to help repolarize and restore homeostasis to the membrane. Now, of course, most of my diagrams are pretty old school. The Na1, K1, ATPase, or sodium-potassium pump is actually a complicated protein structure in the membrane that generates an electrochemical gradient for sodium and potassium. As a result, it's capable of exchanging 3 sodium ions, that is, booting 3 sodium ions out and pulling 2 potassium ions in. As a result, our sodium-potassium pump, in effect, decreases the concentration of sodium inside the neuron, and it increases the concentration of potassium inside the neuron. And it also, for each cycle of ATP hydrolysis, decreases the number of positively charged ions inside the neuron by 1. This combination of the efflux of potassium together with the sodium-potassium pump means that trailing behind the action potential is this dynamic repolarization mechanism that keeps the action potential going in only one direction and restores homeostasis to the membrane following that action potential. At this point, then, we've determined what it is that restores the homeostasis resting potential to any given part of our membrane, and why it is that action potentials travel in one direction and not in both directions. We still have 2 more puzzles to solve from that original diagram. Namely, what determines axonal action potential conduction velocity, and what determines the firing rate for a given neuron. The speed of a localized depolarization, how quickly it depolarizes, and its ability to spread spatially via ionic cascade both affect axonal conduction velocity of an action potential. Neuroscientists often call this local current. 2 factors determine local current. First, the electrical resistance of the membrane, that is, its external resistance, and the ease of spatial recruitment in the axon, i.e. its internal capacitance. External resistance depends upon the number of channels in a given area of an axon membrane. Internal capacitance is determined largely by axon diameter. Thus, neurons evolve 2 solutions for faster conductance. Generally, large diameter axons have low internal capacitance and can conduct faster. Many invertebrates evolved to leverage this strategy. In contrast, most mammalian brains rely primarily upon glial cells encasing axons, thereby decreasing channel density at focal points and increasing the reach of local current. In myelinated axons, depolarizations hop, as it were, from one node of Ranvier to another using saltatory conduction, and thereby increasing conduction speed by up to 10 times. Whatever the axonal action potential conductance speed, when the action potential reaches the axon end bulb, it has arrived at the synapse. The depolarization will open voltage-gated calcium channels. Calcium influx will then lead to neurotransmitter release by facilitating the fusion of vesicles to the presynaptic membrane of the axon end bulb, allowing them to release their neurotransmitter payloads. Let's end this lecture module by recapitulating the major steps of an action potential for our typical neuron. Along the dendritic branches and cell bodies, there are many chemical synapses that contain neurotransmitter ligand-gated ion channels. These channels, together with the voltage-sensitive sodium channels, create a graded EPSP, or excitatory postsynaptic potential, that gets summed by the neuron soma or cell body at the axon hillock. When that sum causes a depolarization of at least 10 millivolts, it triggers an all or nothing action potential, a dramatic shift from resting potential of negative 65 millivolts up to positive 40 millivolts. This action potential travels down the axon until it reaches the terminal end bulbs, at which point it causes the release of neurotransmitters.